talk today is on metagenomic next generation sequencing for infectious disease diagnosis and management. We have two terrific speakers, Chaz Langelier, I always get the wrong. Langelier is Associate Professor of Medicine in our Division of Infectious Disease here at UCSF Health, and Assistant Medical Director of Hospital Epidemiology and Infection Prevention. He's attending on the ID service, and his research really bridges basic science and clinical medicine through studying advanced techniques to diagnose infectious disease. He's become a national and actually world leader in that effort. The co-presenter will be Natasha Spottiswoode, who is Assistant Professor in the ID Division. She attends on both the general and transplant ID services. She's a physician scientist who studies many of the same issues, but with the particular focus, again, looking at advanced techniques to better and earlier diagnose infectious diseases. We're thrilled to have both of them here, in part through the leadership of Charles Chu, who's here. We've really become a national and international leader in this really important and evolving science. So looking forward to hearing both of you.
All right, well, thanks so much for that introduction, Bob, and thanks so much for the invitation today. Well, let's start things off with a case, and this is a case of a 58-year-old woman with HIV who was on antiretroviral therapy, the CD4 count of 165, and presented to the ED with 5 days of fever, cough, diarrhea, and shortness of breath. And her symptoms began while on vacation in Las Vegas. In the emergency department, she was tachycardic, tachypnic. Her O2 sat was 72%. She had a temperature of 38.9 and a white blood cell count of 21. Her chest X-ray demonstrated extensive bilateral opacities. Started on broad-spectrum antibiotics and vasopressors. By hospital day 4, she remained in persistent respiratory failure. However, she started showing some improvements in terms of her leukocytosis and shock. And a number of infectious disease diagnostic tests had come back, although many were still pending. A respiratory virus PCR was negative, blood cultures were no growth, tracheal aspirate culture just showed some mixed gram-positive flora, cryptococcal antigen was negative, and then Coxy, histo, and AFB studies were largely still pending. But one infectious disease diagnostic test did come back positive that really changed management, and that was plasma metagenomic next-gen sequencing, or mNGS. And it demonstrated Legionella pneumophila, a notorious respiratory pathogen that can lead to sepsis and ARDS. And really the topic of our conversation today will be about metagenomic next-generation sequencing. But before we get into that, just some brief follow-up. Because of this result, antibiotics were able to be narrowed to levofloxacin alone. Ultimately, the tracheal aspirate PCR test that was done came back positive for Legionella. And fortunately, the patient fully recovered and was discharged 3 weeks later. So over the next 45 or 50 minutes, we're going to go through 5 key objectives. I'm going to turn things over to Dr. Spottiswoode, who's going to provide some background on metagenomic next-gen sequencing, discuss our current clinical use of the technology. Then I'll talk about how post-immune response profiling can complement detecting microbes by metagenomics, talk about research and development for respiratory metagenomics, and then finally finish off with a discussion about epidemiologic surveillance. So with that, I'll turn things over to Dr. Spottiswoode.
Excellent. Thank you so much, Chaz, and thank you, Bob. So I'm going to start by talking about mNGS in the armamentarium of infectious disease diagnostics. And I'll first start by saying what mNGS is not. mNGS is not culture. Culture is really the first diagnostic technique that helped us understand, move beyond a syndrome to a pathogen. It was developed in the 1880s by Louis Pasteur, shown here with an early iteration of media for culture. And it's still largely the current gold standard for bacterial and fungal infection. Now, while culture has served us amazingly well, we still have many infectious syndromes for which we don't usually find the pathogen. So in a large-scale US study of pneumonia, the authors estimated that over 60% of cases never have a defined etiological pathogen. In sepsis, depending on the population studied, the rate of culture-negative sepsis or sepsis without a clear cause is somewhere between 1/3 to 90%. Super high. And in meningitis, there's a similar problem where 15 to 60% of meningitis cases don't have an identified pathogen. And without knowing what we're treating, we're often treating people with antibiotics that are not targeted towards the bug that's making them sick and may also be overly broad. So I think about infectious disease diagnostics on a scale between unbiased and biased. And what I mean by this is to what extent the answer you get out of a test depends on the question you're asking. So single pathogen tests are by definition biased. A good example is a SARS-CoV-2 PCR. If a patient comes in very ill, as Chaz's patient at the beginning did, to our emergency room, and we send SARS-CoV-2 testing, we can understand if that patient has COVID-19. But that test does not tell us if they have RSV, influenza, any of the many circulating respiratory pathogens that are currently sort of plaguing us all. Similarly, if someone comes in with elevated liver function tests, a Hep C antibody tells you about hepatitis C. They're biased because you have to ask the right question to get an answer. An unbiased test answers the question, are there any pathogens and what are they? mNGS is not perfectly unbiased, but it is the closest thing that we have to an unbiased test because in theory, it will give you any genetic material in a sample that is non-human, whether that's bacterial, fungal, viral, or eukaryotic. In the middle here are culture and pathology, our old friend culture. I used to think of culture as an unbiased diagnostic because when I send a blood culture, I'm not thinking of a specific pathogen. But by sending a blood culture and expecting to get an answer, you're presupposing that there is a bacterial or fungal pathogen that can grow, that it's not something fastidious like Legionella or a virus or a parasite. Similarly, to get the answer of a pathogen from pathology, you really have to know what it is that you're looking for. So focusing in on mNGS, I'm going to take you through the basic workflow. You start with collecting a sample at UCSF right now for clinical use. That's either CSF or plasma, but in theory, it could be anything. And there are studies of bronchiolar lavage, tracheal aspirates, abscesses, and more. You extract DNA and RNA, and you perform random fragmenting, barcoding, and unbiased amplification. So everything is amplified and then sequenced. And then you compare it against a genomic database, subtract out the human, and what you're left with is everything there that is not human, agnostic to what it is. So let's change gears and talk a little bit about the current clinical use of plasma mNGS. And if you'll indulge me, I'll take one diversion on the way there, which is to talk about clinical mNGS for CSF. I feel I have to talk about this. This is how clinical mNGS really started. By the amazing work of Michael Wilson, Dr. Charles Tu, and Dr. Joe DeRisi, they were able to seriously contribute to the problem of undiagnosed CNS infections. They created an RNA and DNA CSF metagenomic next-gen assay, which was launched in 2017, and it has really impressive positive and negative predictive value for CSF infections. This test was developed at UCSF, but it did not stay there. It's gone on to be used across the United States and worldwide, as shown in this graph and map. And CSF mNGS has really established itself as a test that can make absolutely key diagnoses. In a recently published 7-year performance analysis of the CSF mNGS testing at UCSF, they were able to show over 4,828 samples, a wide variety of pathogens, and in 1,000 patients where there was available clinical and microdata, they showed that in many of the diagnoses that were infectious, mNGS was either the only test that made the diagnosis or it made it first. And these included such key diagnoses as zoonotic RNA viruses, hard to detect bacteria such as MTB, fungi, parasites such as the brain-eating amoeba, acanthamoeba, DNA viruses, and more. So acknowledging that CSF mNGS has been extremely successful, I'm now going to talk about plasma. This is the Department of Medicine. And so this is the test that we most often use. And I think the test about which there is some more uncertainty and lack of clarity about how and when we should use it. So let's just talk through the basics. UCSF, until a year ago, contracted with Carius Incorporated for plasma mNGS. In January of 2025, so almost exactly a year ago, we switched over to an internal assay that was developed in the clinical microbiology lab. And the other thing I want to say that's different from CSF is that both Carius and our currently used assay are DNA only, meaning that they do not detect pathogens such as some viruses that are only RNA. Now, what literature do we have to guide us on the use of plasma mNGS? Well, the literature looks a bit like this pyramid. There are a small number of multi-center syndrome-based observational studies that give us a sense of utility in specific syndromes. There are many single-center experience studies, which, while interesting, are less helpful because they're often looking at different populations, use the test in different ways. And then there are really no shortage of very impressive case studies where mNGS has enabled a diagnosis by itself or been the earliest test and changed management. Again, impressive, but not helpful when we consider how we should use this on a broad scale. You may notice also something that's missing from this pyramid, which is randomized control trials. There have not been any high-quality randomized control trials of mNGS as a diagnostic, although that will change soon. So answering the question of how should we use plasma mNGS, I've mentioned it's an area of active investigation. For further context, I would say that given the historically high cost of this test, much more expensive than other infectious diagnostics, and a relatively long turnaround time, many centers have used this as a last resort test to really only be used if everything else has been unrevealing and they still don't know what's wrong with the patient. UCSF stands out as a relatively high mNGS utilizing center. We send more tests, we tend to send them earlier. And so approximately a year ago, the UCSF infectious disease team was really tasked with developing internal use guidelines. When should we use this? How should we use this? So to talk through how we made those guidelines, I'm going to briefly review the evidence for 4 mNGS use cases: severely immunocompromised patients with pneumonia, heme malignancy patients with febrile neutropenia, culture-negative endocarditis, and critically ill patients with sepsis. So starting with immunocompromised patients with pneumonia. One of the key studies here is called PICkup. It was a multi-center prospective observational study that compared plasma mNGS to standard of care diagnostics in immunocompromised adults. I will say one of the centers was here, and one of the local leaders of that study is Dr. Fung in the audience. All these patients had pneumonia of unclear etiology, and they were all profoundly immunocompromised with either a stem cell transplant and/or heme malignancy. These patients got usual diagnostics. They all got bronchoscopied, and all of them had Carius done retrospectively. And then a panel of physicians retrospectively determined the diagnosis. Now, only about a third of these patients got a diagnosis through usual care, including bronchoscopy, which seems low, but jives with the very low rates of pneumonia diagnosis that I presented earlier. 42 got a diagnosis through a composite of mNGS and usual care. And of the 12% of patients who were exclusively diagnosed by mNGS, that included some very key diagnoses such as pneumocystis jirovecii or PJP, or previously called PCP, agents of invasive fungal infections such as mucormycosis, and multiple cases of fastidious and virulent bacteria such as Legionella, again, thinking back to that case that Chaz gave at the beginning. They estimate that about 10% of these diagnoses might have changed management. I do want to say that they also identified some organisms of less clear significance. Also on this graph is human beta herpes virus 6, which does reactivate in critically ill patients and is not generally thought of as a cause of pneumonia, as well as several oral flora of sort of unclear virulence, including Prevotella and Rothia. It's also important to me that we talk about what mNGS misses in addition to the amazing diagnoses it enables. So in this study, 14.5% of patients had positive diagnostics by usual care, including bronchoscopy, but negative Carius testing. These included 7 patients with bacterial pathogens that were all common respiratory bacterial. All the patients with pneumonia due to an RNA virus were missed because, again, this is a DNA-only test, so it will not pick up rhinovirus, SARS-CoV-2, parainfluenza, and a lot of other respiratory viruses. But perhaps most concerningly to me, it also missed 17 cases of fungal infection, including 16 of aspergillus. And there are other literature that suggests it might have lower sensitivity for pulmonary aspergillosis in particular. Changing gears, I'm now going to talk about heme malignancy patients with febrile neutropenia. A brief word on febrile neutropenia. Febrile neutropenia is a syndrome of concern for infections when elevated temperature coupled with really profound immunocompromise. So at UCSF, it's ANC less than 500 or less than 1,000 and falling, and high-risk neutropenia is really encountered in the settings of cancer, chemo, and or a bone marrow transplant. Now, most patients with this are on prophylaxis of antibiotic prophylaxis already when they're neutropenic and before they develop a fever. So partially as a result, most patients don't get a microbiological diagnosis. It's estimated at 20 to 30%, which if you look at it the other way, means that 70 or to 80% of patients are never diagnosed. There have been some Carius studies on this. One of them took 55 patients, and they excluded 7 who either had no infection or an RNA virus. And of the remaining 48, 10 of them, or about 20%, had positive blood cultures, and a whopping 41 or 85% had positive Carius. It is worth saying that after adjudication with a physician panel, they determined that while 12 of those cases were sort of definitive diagnoses, meaning that they matched with the blood cultures if positive, or if not, they were definitively causing the fever, many of them were these sort of probable or possible pathogens or microbes of unclear significance. Oral flora, which is not super virulent, reactivating DNA viruses of unclear etiology, of unclear significance. And there were 7 misses as well. They estimate that antimicrobial management might have changed in about half of these cases, but that does assume that you would be able to de-escalate antibiotics, say from cefepime, based on the absence of a bug like Pseudomonas. Moving on, I'm going to talk about endocarditis. Endocarditis for me is a sort of clear use for mNGS in that you're looking at a pathogen that has by definition infiltrated the circulatory system, so we expect circulating microbial DNA. And mNGS does indeed perform well for endocarditis overall. This is a study of 23 patients with infective endocarditis. 22 had positive blood cultures and 22 had positive metagenomic next-gen. mNGS was very concordant with culture and also was detected longer into the course, which makes sense because blood culture relies on having live bacteria that you can grow. And if you give someone antibiotics, you might kill off those bacteria, make the blood cultures negative, but still be able to detect microbial DNA. I will say, for culture-positive endocarditis, culture gives us the sensitivities. It is currently faster and it is currently cheaper. So I don't see this as a replacement at this time. But where it really shines is culture-negative endocarditis. So thinking about what culture-negative endocarditis is, culture-negative endocarditis can happen if you have a pathogen that is dead from prior antibiotics, if you have a pathogen that's hard to culture. A lot of indolent causes of endocarditis are mouth bacteria that may not grow in culture. There's also a number of zoonotic or environmental pathogens that can cause endocarditis that really don't grow in culture, so Legionella being another example, but in this population, especially Bartonella, Coxiella, T. whippley. And also, if the pathogen is not bacterial, so Candida endocarditis is a big problem. Fungi are often not grown on blood cultures. Of course, you can also have apparent culture-negative endocarditis if there is no pathogen, and you're dealing with a non-infectious mimic such as valvular degeneration, a thrombus, or a malignancy. Now, there have been small case series looking at some common causes or relatively common causes of culture-negative endocarditis, and those case series show very good sensitivity of mNGS for things like Bartonella, which is a relatively frequent cause of endocarditis that we see. There's also been a very large-scale UCLA study that suggests that one of the highest utility uses for mNGS is cases of culture-negative endocarditis. I'm going to touch on one last use case before going over our guidelines, and that's critically ill patients with sepsis. This is something that our lab has done a ton of work on, along with many of the people in this audience. And what we've shown is that our experimental assay, which is distinct from Carius and the assay we're using now in clinical work, is very good at measuring sepsis. It's very sensitive for common sepsis pathogens. So this graph is looking at the sensitivity of metagenomic next-gen for sepsis due to culture-confirmed bloodstream infection for different organisms. It's also specific, meaning that it's usually negative in patients who have non-infectious SIRS syndrome. Finally, it also measured a possible pathogen in nearly half of patients with culture-negative sepsis, which, as I mentioned before, is a really big and hard-to-treat group. So very promising. Before I give you the guidelines, I want to talk about the challenges of mNGS testing as someone who is a proponent, but who thinks that there are limitations as well. There are specificity issues. Like any test that involves venipuncture, you can pick up skin organisms like Staph epidermidis, C. acnes, and less commonly, you can pick up lab contaminants like environmental bacteria. But the bigger specificity issue is things that are biologically present, but of unclear relevance. So in someone with mucositis or say severe C. diff, you can detect GI or oral flora at low levels in their plasma, but that does not mean that those are causing the infectious syndrome. You can also often detect reactivating DNA viruses that are of unclear significance, something like HHV6 in a patient who's not that immunocompromised. And so based on those results, there is a real risk of inappropriate antimicrobial escalation. It's one of the reasons why if I'm asked if this test were incredibly cheap, would I send it on everyone? I will say no, at least at this moment. A bigger issue, perhaps, is sensitivity. We don't really know the sensitivity of this test for different infections at different body sites. We have some sense of how it performs for some syndromes, but really there's a lot more to do before we can make confident clinical decisions based on this test. I will say at the moment, I worry about antimicrobial de-escalation based on negative tests or procedures delayed because we simply don't know how good it is. The last limitations I'll mention are cost. Cost is coming down for sequencing overall and for this test as well. It's hard to get exact numbers, but it is still probably more expensive than most diagnostic tests. And finally, turnaround time. This is improved as well since it's in-house. The turnaround of the actual assay is really fast. It can be done in under 24 hours, but at the moment things are batched and done several times a week. So the turnaround time from order to result is usually more like 2 to 3 days. So based on all that, I'll say the indications that we've come up with, and then the work that we're doing to iterate upon these recommendations. At UCSF, we have 2 first-line indications for plasma mNGS use. First line being send them as soon as the syndrome is recognized. The first is severely immunocompromised patients with pneumonia or pulmonary infection, and specifically high concern for an atypical infection such as invasive fungal infection. And we've opted for a relatively narrow definition of severe immunocompromised, which is detailed below. We also recommend sending it in those patients if they're not responding to standard of care. The second is fulminant infection and very strong epidemiological concern for a hard to detect or atypical infection. So that's tuberculosis, Legionella, nocardia, other zoonotics, or invasive fungal infection. We've developed 4 second-line indications, which include culture-negative endocarditis, which is by definition with negative blood cultures and antibodies, fever of unknown origin after negative first-line workup, including blood cultures, imaging, and other applicable workup. Deep-seated lesions or abscesses if you have negative blood cultures and you either can't sample the site or sampling it is unrevealing. And lastly, severely immunocompromised patients with febrile neutropenia with negative blood cultures and unrevealing cross-sectional imaging. We worked with Dr. Chu in the microlab to change the ordering workflow such that it now aligns with our guidelines. We absolutely recognize that this does not cover every possible scenario. So like a restricted antimicrobial, there is an other box where you can say this is another use case applies here, but you just need to put in an ID consult attending name for that patient. We recognize that this is a start and that it will change as we learn more. So we are working to assess the clinical utility of mNGS at UCSF. We are identifying microbes, patient factors, and clinical scenarios that predict high or low plasma mNGS utility. And we are also working to understand plasma mNGS diagnostic performance in key syndromes and for key organisms such as bacteremia, pneumonia, or invasive fungal infection. And finally, we're working to compare the test characteristics of the in-house assay to the established commercial Carius testing on which virtually all the literature is based. Target sample size for this is 1,000 patients. We're making good progress. And as a first look at our data, we've looked at clinical impact of plasma mNGS by organism. So this graph looks at organisms on the y-axis and clinical impact on the x-axis, with the positive side being that way, meaning it changed patient care for the good, negative or neutral in that direction. And what you can see is that the highest impact organisms, so blue is bacteria, purple is fungi, are either hard to detect bacteria such as Bartonella, Nocardia. Legionella is also there, although we've only had a few cases so far. Or agents of invasive fungal infections such as PJP or mucormycosis. I anticipate that as we learn more from our own work and from what emerges in the literature, that we will keep working with micro to iterate the guidelines for this test use and take the best advantage for this exciting technology for UCSF. Thanks so much. I'm going to hand back to Chaz.
All right, thanks for taking us through that, Tosh. Okay, let's move on to talk about host response profiling. And I'd like to start out this section by building on an important point that Dr. Spottiswoode made, and that's with respect to the specificity issue of plasma metagenomic testing, at least the commercially available testing. It has a tendency to pick up incidental microbial DNA circulating in the bloodstream. In fact, if you look at the original 2019 Nature Microbiology paper that described the Carius test, the specificity versus blood culture was 40%. That's a lot of false positives. To put some clinical context to this, we can consider a case of a lung transplant recipient who was hospitalized for an acute respiratory illness. And at the time of hospitalization, they had both plasma NGS and blood culture sent. Ultimately, this person was diagnosed with acute cellular rejection, a clear non-infectious etiology explaining their acute respiratory illness. Yet their plasma NGS test had 5 different organisms detected. You may look at them and say, oh, well, they're common commensals, but you can appreciate how something like this could lead to antibiotic overuse, especially for folks who might not be that familiar with these somewhat esoteric microbes in some cases. So what do we do about this issue? Well, it turns out that metagenomics actually has a built-in feature that allows us to address the specificity issue. And this built-in feature is the ability to simultaneously profile the host immune response when RNA is sequenced. And why does that work? Well, it's simple. It's because both humans and microbes share the same genetic code. And if you think about most infectious diseases and most infectious disease syndromes, they're not just about a microbe in isolation, but really they represent a dynamic relationship between host and pathogen. So you can probably appreciate how valuable it could be to have information about whether or not there's an immune response consistent with infection if a given microbe is detected by metagenomics, or 5 microbes in that, for instance. So let's focus in on sepsis for a moment. Sepsis, it turns out, is defined as a life-threatening organ dysfunction caused by a dysregulated host response to infection. So it's a good example for understanding how host response profiling can be beneficial for metagenomic diagnosis. And to do that, I'd like to share with you some results of a study that we did in collaboration with Carolyn Kelfie, Michael Mathey, and others. And in this study, we sought to define a host response signature that distinguished sepsis from non-infectious systemic inflammatory illnesses in critically ill patients. And we leveraged the early cohort, a prospective observational cohort of patients admitted from the emergency department to the ICU at UCSF and San Francisco General. And from patients enrolled in this cohort, RNA and DNA metagenomic sequencing data were generated from whole blood and plasma. And importantly, a multi-physician retrospective adjudication process was carried out to identify those patients who definitely had sepsis, met by a clinical definition, as well as microbiologically confirmed, or patients who definitely did not have sepsis and had a clear non-infectious explanation for their illness. And then we asked what was different with respect to host immune response by looking at the gene expression data obtained from RNA metagenomic sequencing. And what you're seeing here on the heat map on the side is really a profound difference in the transcriptional profile in the blood of patients with sepsis versus those with non-infectious inflammatory illnesses. Over 5,800 genes were differentially expressed. That's more than a quarter of the human transcriptome. On the right, you can see genes that are upregulated in sepsis. They're shown in red. The top 50 genes are highlighted here on the heat map, and we have genes such as those important for neutrophil activation. Genes downregulated in sepsis, examples include genes important for T-cell activation. Well, we can then take this really distinct transcriptional signature and apply machine learning methods to distill down all of these genes to just a few that are highly predictive of whether or not someone has sepsis or someone has non-infectious systemic inflammatory response syndrome when they're admitted to the ICU. We can train a machine learning model on those data, and we can build a diagnostic classifier. And in fact, it performs really quite well. And we can assess performance with cross-validation. And we observed an area under the curve of 0.9, demonstrating very high accuracy for distinguishing these two conditions. So how can this be applied to informing the significance of a microbial metagenomic result? Well, we can overlay that classifier sepsis probability output against the microbes detected by plasma metagenomics. So here on the y-axis, you're looking at a sepsis probability from 0 to 1, the output from the diagnostic classifier. And then along the x-axis, you're looking at the different microbes detected by plasma metagenomic sequencing. And here we're focused in on really the most clear-cut cases of sepsis, those with blood culture confirmed sepsis. You can see that there were a few patients in whom plasma metagenomic sequencing did not identify the pathogen picked up by culture, but in those cases, the host response was really overwhelmingly positive and consistent with sepsis, highlighting its utility in those false negative cases. We can also take a look at the flip side and take a look at those patients who definitely did not have sepsis And you can see that in a number of these individuals, metagenomic sequencing of plasma actually picked up microbes that were clinically insignificant. But in only one of those cases, the one on the far left, an E. coli case, was there actually a host response signature that was suggestive of sepsis. Turns out that we went back and took a look at that case. It was actually someone with diabetic ketoacidosis, possibly may have had an infectious trigger. So with that last plot, it really highlights how bringing in host response can improve the specificity of a plasma metagenomic assay, address this key issue that currently is a major limitation. You may be wondering what happened in the culture negative sepsis cases. Well, 74% were positive by the host score, and over half had a possible pathogen detected by metagenomics. Well, combining host response profiling with microbial metagenomics has value far beyond sepsis. In fact, a lot of the work in my lab over the past several years has focused on deploying this technology to improve the diagnosis of pneumonia. And really fantastic work in Michael Wilson's lab in neurology has demonstrated how combining host and microbe metagenomics can improve the diagnosis of TB meningitis. And then really fantastic work in Charles Chu lab has extended this to all-cause meningitis and really done something that hasn't been done before, and that is bring host microbe metagenomics towards a clinical assay, which is undergoing validation right now, and really has outstanding performance with an accuracy of 0.9 for all-cause meningitis diagnosis. Okay, well, let's pivot now to focus on respiratory metagenomics, research and development. And I'd like to start this section with this photo, which is a photo taken during the early days of the COVID pandemic. And I doubt that any of us will forget the stress, the uncertainty, the chaos of that time. But many of you may not remember how the novel coronavirus that led to the pandemic was actually identified. It turns out it was identified by metagenomics of BAL fluid from a patient who was part of a pneumonia cluster in Wuhan, China. And of course, it turns out that metagenomics was being used to study respiratory infections long before the COVID pandemic. In fact, UCSF has been a leader in this area for over 15 years, starting with work in the Joe DeRisi lab in the Department of Biochemistry and Biophysics, focused on using metagenomics for viral pathogen discovery, work that was carried on by Charles Chu, work that was carried on in my lab and by others. So these days, due to advancements in bioinformatics, detecting respiratory viruses is relatively straightforward. However, when it comes to detecting bacterial and fungal pneumonia pathogens, things are a different story. In particular, there's a challenge distinguishing bacterial pathogens from commensals that are present in the respiratory tract. And why is this? Well, the reason is because unlike CSF or plasma, theoretically sterile site fluids, there's the presence of a background microbiome in the lungs that really complicates bacterial pathogen detection. So everyone in the room right now has thousands of different bacterial species living happily in their lungs, whether you're healthy, hopefully, or sick. And these are all picked up by metagenomics. So how do we deal with this issue? Well, it turns out after a lot of work studying patients with true respiratory infections and true pneumonia and comparing them to patients who definitely don't have respiratory infections, we observed some very significant changes in the compositional structure of the lung microbiome that happen during pneumonia that are captured only by metagenomics that allow us to accurately identify bacterial pneumonia pathogens. And those two things are rather simple: collapse of the normal diversity in the lung microbiome and dominance of respiratory pathogens. So what does this look like graphically? Well, we can pull up a plot that on the y-axis shows the abundance of microbial DNA or microbial reads per million detected by metagenomics. And on the x-axis, you're looking at the rank by abundance. So this is a patient who has pneumonia due to Haemophilus influenza. And what you can appreciate if we just look at the top 10 to 15 microbes is that there's really one microbe that's dominant in the airway of this patient. This is BAL fluid metagenomic sequencing. And that microbe is Haemophilus, the pathogen causing the problem. The same type of pattern exists across bacterial pneumonia and at least for several types of fungal pneumonia. And we can see that here in a patient with pneumocystis pneumonia. Pneumocystis is really dominant in the airway. Oh, but they have a low amount of rhinovirus A that's also there as a co-infection. So it turns out that we can build computational models that can recognize these changes and accurately identify pathogens from respiratory metagenomic data, positioning us to be able to have a viable and useful diagnostic test. So let me now share with you some data on how respiratory metagenomics could work from an observational cohort study that we did right here at UCSF in collaboration with Carolyn Calfee and Joe DeRisi. We looked at 92 patients with acute respiratory failure who were admitted to ICUs at Moffitt or San Francisco General. And in this cohort, patients underwent adjudication, just like in the sepsis study, to have definite pneumonia. We identified those folks, and we had patients who clearly had non-infectious causes as well. I'll point out that standard clinical testing in the patients who met a clinical definition of pneumonia only identified pathogens in 43% of cases, consistent with the data that Dr. Spottiswoode showed at the beginning. Importantly, metagenomics identified missed pathogens in 62% of those cases with negative testing. These included common pathogens like Streptococcus pneumonia, which can be killed with a single dose or a few doses of antibiotics, as well as less common pathogens like histoplasma, and potential pathogens that cause pneumonia in certain immune-compromised populations like coronavirus HKU1 that can't be detected by assays currently available at UCSF. Importantly, we found that in those microbiologically confirmed cases, metagenomics had 95% accuracy compared to the gold standard diagnostics that we know and love at present. So what is the status of clinical respiratory metagenomics at UCSF right now? Well, right now it's only available for research and for hospital epi and infection prevention. But that is changing thanks to work in the Chu laboratory, respiratory viral metagenomics. An assay has been FDA validated and is poised for deployment. An assay that can detect everything from rhinovirus to measles. And clinical validation and testing of a bacterial fungal metagenomic assay, plus or minus host response, is in progress and underway. So stay tuned. All right, let's move on to the final section, which is metagenomics as a tool for epidemiologic surveillance. So there are a number of features that make metagenomics particularly valuable for epi surveillance. Number one, it offers broad range pathogen screening in a single test. So you don't have to design your study to have 50 different diagnostic assays to figure out what's causing pneumonia in a certain part of the world. You can have one test that will get most of them, if not all of them. It offers the opportunity to identify uncommon or novel pathogens for which diagnostic tests may not exist. This happens not infrequently in epi surveillance studies with metagenomics. And there's actually no need to maintain pathogen viability in culture following sampling or have comprehensive on-site microbiology lab infrastructure in a remote part of the world. Samples can be put in a preservative that stabilizes nucleic acid, brought to a central city, processed and analyzed there. So let me just provide an example of how this can work from a study that we carried out a couple of years ago with Lawrence Wong in pulmonary and William Barodria, who's a professor at Makerere University in Kampala, Uganda. So this study sought to understand the etiology of community-acquired pneumonia in Ugandan adults living with HIV. We studied 217 adults who were hospitalized for pneumonia at the main tertiary care hospital in the country. We carried out RNA metagenomic sequencing on BAL fluid. And there were a number of interesting findings from this study. Number one, a potential etiologic pathogen was identified in 97% of cases. And you can see from the pie chart here that in 52% of those cases, that's the colored slices, an established respiratory pathogen was identified. But in 45% of cases, a possible but uncommon respiratory pathogen was picked up. So what were the common established pathogens? Well, we can just take a look at the top 10 here, and we can appreciate that, perhaps not surprisingly, Mycobacterium tuberculosis was number one, and a number of other potentially expected respiratory pathogens like pneumocystis, Haemophilus, influenza A, metapneumovirus were picked up. One thing that surprised us, however, was the fraction of cases that were present as co-infections. In particular, we found that 38% of TB cases were actually found as co-infections with another established pathogen. And we don't always think about that. Often if someone has a diagnosis of TB, we think that that is all that's going on and what brought them in. Not the case in this study. You may be wondering, what were those uncommon pathogens? Well, there are a number of interesting ones that were picked up that were unexpected, like rubella virus or T. whipplei. So just an example of how metagenomics can be useful for epi-surveillance. Of course, metagenomics can be used for epi-surveillance studies, not just for pneumonia, but also for other infectious syndromes like sepsis. I have to tell you about a really exciting surveillance study that's currently underway to understand the etiology of sepsis in the United States. And this study is evaluating 1,500 patients from 60 hospitals across the US. It's a collaboration with Carolyn Calfee, Michael Mattei, Nate Shapiro at the Beth Israel, and other individuals as well. And this study will leverage plasma collected within 4 hours of sepsis onset, leveraged from a clinical trial of fluid management for sepsis that was recently carried out. Importantly, I'll point out that standard clinical testing identified a pathogen in only 30% of cases, so inevitably metagenomics is going to add value here in terms of shedding light on what causes sepsis in our country. So stay tuned for the results of this study. Okay, well, let's move on to the final section of our talk today, and that's metagenomics for hospital epidemiology and infection prevention. And I have to say that UCSF has really been a leader in the precision infection control space. And one example of this was the development of the UCSF Genomic Epidemiology Rapid Response Program, which was established in 2017 with the goal of tracking and mitigating hospital onset infections to improve patient safety using advanced genomic approaches. This was one of the first programs of its kind at an academic medical center in the world, and the first program to ever implement routine use of metagenomics for hospital epidemiology and infection prevention. To date, there have been over 80 investigations, not just at UCSF, but also at Stanford, UC Davis, and in collaboration with the US CDC. And I'll point out that the most common outcome was actually rapid rule out of a suspected outbreak in almost 70% of cases, saving valuable time for medical professionals and providing a definitive, confident answer in terms of whether or not a hospital onset infection had taken place. So let's just take a look at an example of one of these investigations to provide a final idea of how metagenomics can be useful in this scenario. And I also should point out that if you're interested in learning more about the program, there's a paper that is coming out this afternoon in the Lancet Microbe describing its development and performance over the past 7 years. So let's move on to a case, one final case for today. And this was an investigation of a septic transfusion reaction that involved a 60-year-old gentleman with acute lymphocytic leukemia who underwent bone marrow transplantation. And they were hospitalized for that, and they had done well following engraftment and were being prepared for discharge. On the day of discharge, it was appreciated that they still had some thrombocytopenia. So the team ordered transfusion of 2 units of platelets before they left the hospital. The first unit, the transfusion of the first unit went just fine, but 15 minutes into the transfusion of the second platelet unit, the patient developed rigors, fevers, and critical hypotension. They were started on broad-spectrum antibiotics, given IV fluids, vasopressors. Eventually, blood cultures came back positive for Acinetobacter. And this obviously was of significant concern, so the hospital epidemiology team took a deeper dive, worked with a clinical microbiology lab and clinical labs to try and understand what was going on. So further investigation demonstrated that actually there had been 2 other co-components that were collected along with the unit of platelets that was transfused into the patient and associated with the subsequent sepsis. The first unit of platelets that didn't have any issues was from a completely different donor. Cultures were done on these 3 platelet units, and only the culture from the unit that was transfused into the patient came back positive. First piece of information. Second thing that was realized was that actually these platelets had undergone a specialized process of, quote, pathogen reduction that involved UV light and a chemical cross-linking agent. And at that time, there had been no failures of this pathogen reduction process in the United States. In part because of this, the blood supplier really felt strongly that there was no way that there could have been a failure of this process, and that instead of the platelets being responsible for the patient's subsequent sepsis, a more likely scenario was that the patient on the day of discharge was actually developing sepsis, and the transfusion just happened to coincide, and that actually the positive culture from the residual platelet bag material was probably backflow from the patient's blood into the platelet bag. Needless to say, we did a deeper dive, and one of the ways we did that was using metagenomics. And so we did metagenomics on a number of residual plasma samples that were available, and the results are shown here on this plot. So the y-axis, you're looking at the amount of Acinetobacter DNA in a sample. We can first focus in on the right side of the plot, and you can appreciate that we had a sample of plasma from the patient's morning labs at 4:52 in the morning. There was no Acinetobacter in that sample. You can see when the transfusion occurred, then you can see the 3 available samples that we had, that we were able to sequence after the transfusion. There was a lot of Acinetobacter. If we look at the platelet products, however, really the transfused platelets contained an exceedingly high level of Acinetobacter. Putting things together in terms of its temporal context is really strongly argued that it was the platelets that had led to the Acinetobacter in the patient's bloodstream and the subsequent sepsis. To really prove this, however, we had to do phylogenetic analysis, which it turned out revealed that the genomes of the Acinetobacter cultured from the residual platelet bag and from the Acinetobacter cultured from the patient were genetically identical, and it just happened to turn out that they were completely novel Acinetobacter species, most closely related to one called Acinetobacter nosocomialis, aptly named. So in terms of follow-up, fortunately, this patient fully recovered. They were discharged 16 days later. And this ultimately led to a collaboration with the US CDC, which eventually linked this case to a multi-state outbreak involving 7 patients and sadly 3 fatalities. And the CDC was able to figure out that this originated from a contaminated blood donation equipment issue, which has since been solved. Okay. So to just close out the epi surveillance part of this talk and finish up the talk, I have to come back to highlighting the utility of metagenomics for pandemic preparedness and just show this snapshot, which highlights the fact that in addition to metagenomics being what allowed us to understand the cause of what caused the COVID pandemic, metagenomics done in the Chu lab right here at UCSF allowed us to identify the first case of SARS-CoV-2 Omicron to enter the United States. And that was in December of 2021. And since that time, there have been a number of emerged or re-emerged pathogens. Things have been relatively less chaotic, of course, in 2020, but still plenty of business on the infectious disease side. We've had Mpox, we've had H5N1 highly pathogenic avian influenza that's currently circulating in a number of animal species. We've had a resurgence of dengue and then a resurgence of measles. So we can ask ourselves what's next in the next decade, or the next year, or the next month. Regardless of what it is, without a doubt, metagenomics will allow us to be better prepared to detect it. Okay, so we can finish with some conclusions. Number one, these are the key take-home points. Metagenomics is an unbiased, culture-independent infectious disease diagnostic assay. CSF metagenomics has established utility for central nervous system infection diagnosis. Plasma metagenomics utility varies by syndrome and pathogen, but really the clear case high value use cases are immune compromised patients with pneumonia and severely ill patients with risk factors for atypical infections. Host response profiling can improve existing metagenomic next-gen sequencing assays. Respiratory metagenomics is useful for pneumonia diagnosis and is on track for clinical deployment. And finally, metagenomics is a valuable tool for epi surveillance, infection prevention, and pandemic preparedness. So we'd like to acknowledge a number of people who played key roles in the work that was presented today and open it up for questions.
Absolutely fascinating. Thank you. Thank you both. We'll go over by a minute or two. We started late. I got to start with this. You said it's not ready for prime time. This will be the first test I do on a patient who comes in the hospital with fever in blank years.
Well, actually,
come close to the mic.
Sorry, yes. So the question was, when will this be the first test that we do on any patient with a fever? Well, I think.
I just want a number.
But
in blank number of years.
How about when we deploy a working host response assay? Would that work?
No.
Okay.
I mean, is this 3 years away, 10 years away, or never away?
I think it is somewhere between 5 years and never for any patient with a fever, because I would think about this as a test with utility in the same way that I think about, say, a CT scan. There is, I don't think, going to be a circumstance where I say every single patient with X criteria should always get a CT of their abdomen if they enter the hospital.
We're almost there, but okay.
But CTs have, all tests have downsides. And I think one thing we think about on ID is what is the possible downside of a test if the test is cheap? And in some cases, such as urine cultures, if you send a urine culture when it's not indicated, there are real downsides. That's even the case with blood cultures. You can get false positives. You can also get false negatives. You can be reassured. You can have the patient rehospitalized with no good reason. So I think all-cause fever, we need some sort of host response to make this clinically useful. Microbe alone, I think, can have too much false positives or false negatives.
Guys, you agree with that?
Yeah, I think so. We could also answer by saying when we have an RCT that gives us more information in terms of its utility for fever. I mean, I send it as a first-line test along with others, but I think one has to be prepared to interpret results that may include false positives.
Perfect.
Yeah.
All right, let's. Before I open up, you were a little cagey on the cost. You said you don't really 500, 5,000, 50,000. What, like the range? Like how much?
I think it's a little hard to say because it depends on how the recompense is working. So it is. I have talked to Micro about this, and the actual number is hard to pin down. It's not deliberately cagey at all.
Okay.
I think what I would say is that Carious testing, I think about it as a similar order of magnitude to a CT, like a chest CT. The current test we expect to be cheaper and the cost to continue to go down as it gets cheaper, so less than that. But an order of magnitude for a CT is useful to me.
Several thousand.
I think for Carious, I think the current test is less than that.
All right, open it up. Say who you are, Rich. Yeah.
Thanks. I enjoyed that. I enjoyed that a lot, Chaz and Nori. When you mentioned the epidemiologic studies, I think more of going all the way toward wastewater, as a beginning thing. But do we even do things like NICU, where we know all of these people or patients essentially get infected with stuff that's in the NICU or ICUs, or you could go on and on, operating rooms. But at one point, would we just scan the environment at UCSF like we do with some hot water outlets, for instance, in a unbiased way, and say this is reaching a threshold at which, if you think in the wastewater we vaccinate, or in the hospital we do something about it?
That's a really great question. Interesting you should ask about the NICU. We actually have a prospective observational study in the NICU that's just starting up, being led by Victoria Chu, who's one of the medical directors for hospital epi and infection prevention. And that study is seeking to sample not only babies in the NICU, several different sites on their bodies serially, but also the environment. and to use metagenomics to try and reconstruct transmission patterns, not only of bacteria, not only of pathogens, but also commensals, viruses, et cetera, to really understand transmission dynamics. So I'm hopeful that that study will help to answer your question and to help us understand the potential value of prospective metagenomics of certain environmental sites to provide an early indication of subsequent hospital infections.
Yeah, I'll throw one more. And Chaz, your host response, it seems like the answer is it's either a bug or not a bug, but didn't tell you anything about the pathogen, or does it give you an indication the host responds in a different way to a bacteria or a fungus, for example?
That's a great question. The host response does differ significantly based on pathogen. For instance, there's a really dramatic difference in the host transcriptional signature between viral infections, which typically have high interferon-related gene expression, versus bacterial pathogens, which have other pathways like TNF-alpha signaling. So certainly the host response assay could be tuned to also provide information on pathogens. And in fact, the CSF host response assay that Dr. Chu is developing does just that for different meningitis pathogens, viral, bacterial, fungal, and even provides a score for autoimmune meningoencephalitis as well.
I guess ready for prime time in that if I get a host response that says this is viral and not bacterial, do I stop antibiotics?
I think we're close. You know, we're in the area where we still need some validation studies to be done, but we certainly are close.
Just one thing to extend that is another thing that we're looking at is can you get prognosis as well as diagnosis from a single sample? So our paper has recently come out showing that you can provide an accurate assessment of who is at highest risk of in-hospital mortality using a combination of microbial and host factors as well from the same sample.
Maybe take one more left.
Yeah, can you Lev with hospital medicine? I was wondering if of your indications for sending the plasma, how many of them are ready for us generalists to order without even talking to you versus ones where you'd still prefer to have an ID consult?
That's a great question. So the question is sort of when should you order this before you call us? Well, I think for general ID, some of those indications are relatively less likely to come up. So our very immunocompromised patients with pneumonia tend to be on SOT, solid organ transplant, or heme malignancy patients, because we're talking about people who are really immunocompromised. There are a few that will be admitted to general medicine, such as people with very decompensated HIV. I think a good use case is culture-negative endocarditis, although I will argue that if you have a patient you think they have culture-negative endocarditis, we would love to be called. I certainly think that this is a test that can and should be sent by generalists. I do think that if you're concerned enough about an unusual source of infection that you're considering sending it, it may be time to consider calling your friendly neighborhood infectious disease docs.
With that, we should quit. Thank you both for this work and a terrific talk.